It is a great pleasure to introduce Sanjit Biswas. Sanjit is an MIT alum who pursued both undergraduate and graduate studies in EECS, and he's a leader in democratization of enterprise technologies and building networks of hard techs with AI approaches to broadly impact how people and organizations navigate and operate. He is CEO and co-founder of Samsara, a company that increases the safety, efficiency, and sustainability of operations around the world. He's been recognized as an MIT Tech Review Innovator Under 35, a technology pioneer by World Economic Forum, and one of Goldman Sachs' 100 most intriguing entrepreneurs, and Glassdoor's 218 top CEO. Yay. He and his wife, Hope, started the Biswas Family Foundation in 2023, and they're committed to supporting groundbreaking research and innovative programs that translate to scalable health benefits worldwide. A really nice fit with HEALS. And they have been some of the earliest supporters of HEALS for their work with the Biswas Fellows program, which has been transformative and enabling us to debut one of our flagship MIT HEALS programs this year. And as the faculty member who gets to run the Biswas Fellows selection process, I can say that Sanjit has not just offered financial support, but also support in sort of how we should be thinking about this program. He took us out to dinner with the fellows last night, and I have to say it was a really cool example of one of those convenings where our fellows had a chance to interact with amazing AI scientists, not just from MIT, but from Harvard as well. And they found connections, so it was cool. To share a little bit about the program, he's going to sort of give the vision of why the foundation was interested in this, and then he's going to introduce our first cohort of fellows. So Sanjit, thank you so much.
All right. Thank you, Angela. It's great to be here. It's great to be back at MIT. I was a graduate student here a little over 20 years ago, and it's really fun to see so many familiar faces still around the institute. It was also fun just to see Paulina speaking. She was actually one of our hosts. We had a research platform that was deployed throughout Cambridge, and I remember visiting her apartment in a snowstorm because I had bricked the software on the device, and she relied on us for internet access. So I'm glad it all worked out. So obviously, she's done great work since then. So what I love about MIT is just the incredible nature of this institution in terms of its talent density and willingness to pursue and really investigate really unique and creative ideas. And it's just such a special place. We could not be more proud to support the Institute philanthropically. A few years ago when we met with Ananta and Sally, they were just kind of beginning to form the HEALS initiative. And they said, you know, we're really curious not just about computation or not just around biology or AI, but at the intersection of all these areas and opening it up because as we heard this morning, over 3/4 of the faculty here are working in areas that are related to HEALS. So they were looking for kind of a way to get the program off the ground. We were just excited. We kind of heard 3 sentences and said, we're in. And then the question became, how can we be most helpful? Where can we be most impactful? And one of the interesting things about this sort of science is the diversity of ideas and how many of these are kind of cross-pollinations of ideas from different disciplines. We heard about oceanography and cell biology, and I'd never really thought about those two together, but it makes total sense. That's the kind of work we want to be able to support. It's kind of high risk, high reward, creative disciplines. And for us, being able to create this fellowship program was a way to attract even more talent to the Institute. So our focus has been on early career postdoc fellows. It's going to be a multi-year program, so it'll be multiple waves or cohorts. And it's been fun to get the program off the ground. So thank you, Angela. Thank you, Caroline, for all the hard work. You know, reviewing all these packets, it takes a lot of effort. A lot of just great faculty members have contributed their time. So for us, we're just proud to support, and I'm also proud to introduce the first cohort of fellows. Okay, so if you guys don't mind, thank you. All right, and what I'll do is just to give you like a one-sentence introduction, and then I think each of you will speak for about a minute, and we'll kind of try to keep it fun. So we'll start with Tom Dillon, who's joining us after completing his PhD at MIT, I think defending next week, is that right? Yeah? And he's going to be doing his fellowship under the supervision of Brian Anthony.
Yeah. Yeah, thanks so much for the introduction. Hi, everyone. Yeah, my name's Tom, and my research looks at using medical imaging and robotics to make cardiovascular surgery safer and more effective. So currently, cardiovascular procedures are guided primarily by 2D X-ray imaging. So that means that surgeons have to guess or infer the 3D position of surgical tools and implants in 3D space. So this can lead to severe complications for the patient. For example, implant leakage or device failure. So with support of the Biswas Fellowship, I'll be using tools from AI and virtual reality to create a more intuitive 3D guidance for the surgeon, with the ultimate goal of reducing intraoperative complications. So these types of more streamlined navigational tools can make these procedures safer and more accessible in a wider range of environments, including those with fewer specialized resources. So I can hand it off.
Great, yeah. And next we have Zanaína Macedo de Silva, who completed her doctoral work in Sao Paulo. She'll be doing a fellowship with Forrest White.
Good morning, everyone. And I'm postdoctoral fellow in Forrest White lab. And I'm working with glioblastoma, which is aggressive brain cancer that has remained without new treatments for at least 25 years. And one of the main reasons for this is the new developed drugs cannot cross the blood-brain barrier. So during my postdoctoral research, I'm going to test a new drug that presents a remarkable capacity to cross the blood-brain barrier. And to understand how this drug is working, we are going to apply omic science. So we will be able to elucidate the phosphoproteome repertoire in both primary and disseminated tumors. With this, we hope to understand, for example, how the mechanisms of resistance works. And in the end, we hope to offer for the patients new treatment options.
Okay, and then on the topic of brains, next we have Adia Rakimzan, who will be working with Laura Lewis.
Hi, everyone. Thanks for the introduction. Yes, my name is Adia, and I'm interested in brain states. We all had these mornings when our brain just won't plug in, leaving us foggy and slow. For most, a good night's sleep fixes it, but for millions living with Alzheimer's disease, depression, or ADHD, this is a permanent daily reality. The problem is that we currently lack practical, non-invasive tools to monitor these states outside of hospital. As a Biswas Fellow, I'm bridging this gap with a wearable system that uses light-based technology called FNIRS to decode brain internal state. By training machine learning models, to identify hemodynamic patterns, we can finally track the transitions from arousal, alertness, to sleep in the real world. We are also exploring the use of simultaneous ultrasound brain stimulation to gently nudge these states back into balance. By combining real-time monitoring with precise neuromodulation, we hope help patients to reclaim their cognitive health and daily lives.
Okay, and then next, Rachel Silverstein, who will be joining us from Harvard. She's going to do a fellowship in the lab of Yunhua Huang.
Hi, I'm Rachel, and I study microbial dark matter using genome language models. So bacteria make up the vast majority of life on Earth, but we still don't know what most of their genes do. And studying bacterial genes has led to extremely innovative and important discoveries, such as CRISPR-Cas systems, which are now widely used for genome editing and therapeutics. But CRISPR was actually originally discovered by careful investigation of these mysterious repeat sequences in bacterial genomes. And these could be seen visually by researchers just by looking at genome sequences. Most patterns in genomes aren't detectable by the human eye, but they are detectable using AI, which is why I'm aiming to develop new AI models, which can hopefully help us figure out more of the functions in these unexplored genomes and hopefully find more important discoveries for human health or just life sciences.
All right, and then last but certainly not least, Konstantin Suanas, who did his PhD at the Harvard-MIT Program for Health Sciences. He's going to be doing a fellowship with Sangeeta Bhatia.
Just like we're here today to have conversations, my research vision focuses on the conversations of our cells and tissues. Communication between cells determines whether your body fights off infection or helps it grow faster. Researchers have largely focused on studying a few protein signals, and these have become powerful therapies. But from literature meta-analyses, 90% of all data focuses on just 10% of signals. So we're largely blind to what most signals mean and which ones can help improve human health. I seek to decipher the intercellular conversations of the human body, then redirect and engineer those conversations to unlock entirely new ways of treating disease. As a Biswas Fellow, I will develop new protein engineering tools to synthesize many more signaling proteins than we could before, measure and study their effects on primary human cells and tissues, and create new AI/ML computational methods to discover the right signal to treat the right disease. I'll begin with viral hepatitis infection, but longer term, I'm optimistic that these Biswas-enabled approaches can apply to many more diseases and even allow us to design and engineer new signals better than what exists in nature.
All right. Right, and congratulations again to the first cohort of Biswas Fellows. We're very excited to support you. I hope you all have a chance to meet them. I think there's some networking events a little bit later. Their work is super interesting, and these are some of the most impressive packets I've ever read. So very, very proud to be backing this program, and then we do have more cohorts that will be rolling out in the coming years. Okay, thank you.
Wow, that was truly impressive. I want to work on all of those projects. I can't wait to get an update on those. Hi, my name is Angela Belcher, and I'm a professor in biological engineering and material science engineering in the Koch Institute, and I'm also the faculty lead for the MIT-Novo Nordisk Artificial Intelligence Postdoctoral Fellows program, and I'm excited to update you on that today. This program has been so much fun to be a part of. We started in the School of Engineering in 2023, and then we've recently partnered with HEALS. This program is really the interface between AI and life science. And what we're looking for in these remarkable postdoctoral fellows are fellows that are deep in both AI and life science, and by coming together can really harness the potential for new directions in health and other aspects of life sciences. So I mentioned before, we launched in 2023. We've had 3 cohorts. We are accepting applications for our 4th cohort right now, and the priority closing date, if you know someone that wants to apply or you want to apply yourself, is April 15th for that. So far we've had 26 fellows in this program and 28 faculty advisors. And because we want to be so deep in both life sciences and the biology and the AI, many of these fellows have mentors across departments. And actually, the faculty involved in this are from 13 different departments, centers, and labs across campus. There's been about 25 publications so far and 15 conference presentations. One of the really important aspects of this is that, well, it's very generously funded by Novo Nordisk, but they didn't come in as just funding partners, they came in as mentoring partners. They're involved in virtual journal clubs with our postdoctoral fellows. So we have monthly journal clubs, and we have virtual ones with researchers and scientists from Novo Nordisk. We have social events. We've been able to visit Novo Nordisk here in Massachusetts, and some of our fellows have gone to some of the international sites as well. So again, we have, the richness of the ability to choose really amazing people from this program. And we're going to have two postdoctoral fellows speak to you today about their journey and what they're studying and what their discoveries are. The kinds of topics that we have been recruiting into are AI for imaging, AI for drug discovery, large data sets and clinical analysis, AIs and sensors for data collections, especially related to brain health, AI and robotics to better understand robot-human interactions, and looking at microbiomes, enviromes, and new pipeline molecules for therapeutics. I think our postdocs have had a really amazing experience, and here's some quotes about the interdisciplinary collaboration between clinicians and biologists has strengthened my interest in translational science and real-world impact. I now see myself not only as a researcher, but also as someone who can help bridge the gap between fundamental technology development and clinical applications. And another of the fellows said it's enabled me to build meaningful professional relationships with researchers across engineering, biology, and medicine. So we have these 3 cohorts so far, and the cohorts are very closely interacting, but interaction between these cohorts is also very important. So it's been amazing to be a part of this. I've learned so much myself, and I've had the opportunity to mentor or co-mentor 2 postdoctoral fellows. I want to thank Novo Nordisk for their generous support and their scientific interactions and relationships, and really helping us mentor and develop the leaders of the next generation between AI and health sciences. So with that, we're really fortunate to have 2 postdocs today. Babak Major is going to be our first speaker, and then Cristina Velasquez is going to speak after that. So I'd like to go ahead and welcome Babak to the stage.
Hello. Thank you. I'm excited to share the work that we've been doing during this fellowship in the Department of Chemical Engineering with the rest of the fellowship. I'm working with Connor Coley and Klaus Jensen in the Department of Chemical Engineering, so just to get started, I will be talking about AI synthesis and the use of automation to discover novel reactions and what relevance that has in medicinal chemistry. So overall, we're looking to automate and accelerate the discovery process in molecular research, but particularly the way that we're doing this is through the development of algorithms, protocols, tools, platforms, everything and anything with machine learning, automation, and informatics, machine learning to kind of drive this automated improved societal outcome that we can get from discovering new molecules. And my PhD was in medicinal chemistry, but I did my undergraduate in chemical engineering, back in chemical engineering. But in my PhD, I learned a lot about organic chemistry, and particularly what is the role of novel reactivity. And what I found to be really interesting is that 1 out of every 4 reactions in the pharmaceutical industry are the amide coupling, which typically is going to click together a carboxylic acid and amine to make the amide bond, which is a very important bond in the pharmaceutical industry and in drug discovery. However, it's somewhat biasing the molecules that we're making. If you imagine this big chemical space, we're kind of only searching this one hyperplane as opposed to the entirety of the space. And so if we can invent new reactions, we might be able to break out of this bias and fine-tune molecules towards where we want to go. And this is what that might look like. So for instance, you might see the typical amide coupling in the top left outcome. However, there are also different reactions that can be formed between an amine and a carboxylic acid, for instance, to direct amination or a benzoxyl formation. These molecules that are formed from the same building blocks just by changing the different reaction conditions to get to a new structure enable us to access a range of pharmacokinetic or pharmacochemical properties. And this is very important because drug discovery is a very multi-objective optimization that we're trying to do. And so by being able to fine-tune and control the structure of the molecule, we can fine-tune the properties and ultimately get to the drug that we're looking for faster. And so in my PhD, I learned a lot about the philosophy behind this, but I want to go back to engineering and think about how can we actually discover these more heuristically, more kind of on demand. Because largely to discover new reaction, we're relying on what is called accelerated serendipity, screening a lot of reaction conditions to enable the reaction of desire to actually be invented. But when we're using computational tools, we might be able to come up with a mechanistic model and combine it with all of these new tools that we have to try and get at that novel reaction mode immediately, find the right reaction conditions right away. And so I had developed this model called the virtual flask in conjunction with a series of mechanistic rules that we had derived from rules from a mechanism textbook, and built this kind of like network system which kind of mimics what you do in the lab. The virtual flask, I did my PhD going into a lab with a physical flask, and so I like this idea of a digital twin here. I had a lot of help so far kind of doing this, particularly combining the model with automation. Some undergrads, Joy, for instance, helped with vision model here, and as well as trying to identify solid and liquid interfaces in vials. And this is some images. I have a lot of these, but part of the robot that you can see, some of the interfaces that we've been building to control the robot. And ultimately, what we're finding is that AI, in conjunction with automation, can really accelerate discovering assay types if you build the platform in a way that makes sense and kind of like allows us to use the data in a driven format. So far, it's been working pretty well. The automation has enabled us to discover or use novel assay types. So for instance, this is a kinetic assay where I permute both the time and order of addition, which allows us to see different product distributions despite having the same chemical conditions in the reaction. These are data points that are very difficult to try and extract when you are doing these by hand. No one wants to go back to lab every 5 minutes, every 20 minutes, and change the reaction permutations. And so this really helps us out to try and at least validate the mechanisms that are being enumerated. And this has led to 2 novel reactions, one opening of a benzoscyl ring and one manic-like aminomethylation, which makes a derivative of tramadol, which I just got my wisdom teeth out, so it's kind of interesting for me. But we didn't want to just stop there, and so we went back to the computer, and now we've been focusing on this advanced cheminformatic modeling. And so just like how we have LLMs for chemistry or for proteins and these linear sequences, as you can imagine, molecules are 2-dimensional. They have rings, they have branches, and so we need more of an advanced kind of like language to actually model these. And so I've been thinking about that, using it for transition state searching. And with undergraduates, we've been working on automating the scale-up process of these novel reactions, because oftentimes the reactions that we are extracting are not going to be amenable to scale up. And so if we can automate both the reaction discovery and the heuristics to scale that reaction to industrial scale, we would be good to go. And so future directions, once we kind of get all the synthesis really hammered down well, is to combine this with biological workflows. Particularly, can we use AI and automation to study pathogenesis? Can we use chemical synthesis to perturb biological systems and try and understand what is happening and recreate these disease states? Ultimately, I have to thank a lot of people for everything that has been done so far, particularly, of course, the MIT-Novo Nordisk Fellowship for all the support, providing a structure to this postdoctoral experience, in addition to the whole Department of Chemical Engineering, Connor, Coley, Klaus Jensen, their groups, just everyone. But thank you. I really appreciate it, and open to questions after. Great, yeah.
Hi, so today I'll be talking to you about what I've been doing in my postdoc. But first I'll tell you a little bit about myself. I'm interested broadly in sleep, but I'm particularly interested in memory and its relationship to sleep and learning. And I work jointly in the labs of Patty Mass and Laura Lewis, with the Mass lab working more on the AI side of things, particularly in the context of using AI for human benefit and enhancing health and well-being, whereas the Lewis lab works more in the life sciences part of things, particularly in understanding sleep mechanisms and developing new ways to study them. And I'm a MIT-Novo Nordisk Artificial Intelligence Fellow. So a little bit of background about the project. It'll focus mostly on slow waves, and these are brain waves that occur at around 1 hertz when brain cells at the surface of the brain oscillate in synchrony, kind of like individuals in a stadium synchronizing to form a Mexican wave. And in health, we know that these are really crucial to the kind of learning and memory benefits that sleep confers. And they are also key drivers in the brain's waste clearance system, also known as the glymphatic system. So it gets rid of metabolites and toxins that otherwise would accumulate there. And we know that as we age, and particularly in people who have dementia or go on to develop dementia, these slow waves become less efficient over time. They become less powerful and less frequent, and this is associated with memory and cognitive deficits, and a buildup of the protein and waste that would otherwise be cleared during sleep. And this, we believe, is associated with the neurodegeneration that is seen in Alzheimer's and other forms of dementia. So one possible solution to this is to enhance said slow waves. And what my lab mate Nathan Whitmore has been working on is an app that runs on a smartwatch, and this app has been trained through an LLM to look at various sleep measures, both using EEG and the sensors available on a smartwatch, so looking at heart rhythm and movement, and using this to automate sleep staging, which would otherwise be quite a laborious task. And when it sleep stages, it can detect when somebody is in slow wave sleep and deliver a stimulus, a sensory stimulus non-invasively. And this has been shown to enhance slow wave power in both younger and older adults. So this provides a really nice way to both study slow wave enhancement and slow wave manipulation in a naturalistic setting in people's homes, but also from like a clinical standpoint is really beneficial because it allows us to do larger scale studies for longer durations of time. So what I'm doing now as part of my postdoc is I'm applying this to older populations who are otherwise healthy, but maybe starting to see these deficits in slow waves. And we're looking at a 2-month intervention period where we will be delivering this intervention in the participants' homes. And we'll be looking at changes in brain structure, changes in glymphatic activity, cognition, mood, before and after the intervention to better understand how this might be able to benefit people who might be starting to see slow wave deterioration due to age. And yeah, we're excited to see what results this study yields. And I finally just want to thank MIT-Novo Nordisk, the Media Lab, and both of the labs that I'm working with, the Fluid Interfaces and Lewis Labs. Thanks.
Great set of stuff, and so you can see in this section too, a core mission, a core goal of MIT is to train that next generation of scientists, engineers, and researchers, and that same is true for MIT HEALS. A core goal that we have is to be able to also train the researchers to come, particularly the grad students. And I think not only is that an important mission of MIT, actually the grad students really are a key driver of our discoveries, of our innovation, of the work that we're doing. And so the ability to leverage that training and that discovery is pretty key. Now more than ever, doing that is a challenging thing. We've talked about this idea of convergence, and so bringing together these different disciplines, these different ways of thinking, doing that for an individual within one lab or within one department can be a limitation at times. And so the core goal of the MIT HEALS Graduate Fellowships is to be able to leverage this and bring people together in new ways. The other thing I would say is actually the grad students are the glue that connects all of us. So most of the collaborations from our lab have actually come from a student in our lab knowing a student in a different lab, them talking over coffee or beer or something like that, and coming together in different ways. And I think that happens really effectively within a graduate program. It actually doesn't happen as well between different graduate programs. And so we can overcome that by basically bringing amazing people together in different ways. I think that not only will have an impact on their research and their labs, but that cascading impact on everybody that they know throughout their different programs. And so this is really the framework that MIT HEALS came to it. And so we've been really fortunate to be able to have an exceptional graduate fellow program over this past year. So we actually received 220 applications for these graduate fellowships. We were able to award 32 this year. These students come from essentially, I think, let me see if I actually have that. Yeah. This is an exceptional group of students. They come from almost every single graduate program at MIT. They represent beautiful work across the spectrum of health and life sciences, from fundamental and molecular up to applied and systems. In addition to providing support for that for 1 year for their fellowship, for their tuition and their stipends, the thing that we actually also get is ability to bring them together. So I've been really lucky to be able to meet with them monthly. I've gotten to hear about their work. They've shared this with each other. I've seen new connections form just on the fly as people have been brought together in that way. And then we're also working to ensure that we can help support their training and their ability to drive forward entrepreneurship or translating their work into a next direction. So I've gotten to hear many of these people present. You are in for a treat. You're going to get to hear 4 of these presentations today. I'm so looking forward to this. And we're going to begin by a great presentation from Diego and Francisco Santos Rivera's lab.
Thank you. Hi. Can everyone hear me? Yeah. Cool. Each of us carries between 4 and 5 million genetic variants that differ from the reference genome. And we'll continue to acquire more as our cells divide over time. Most of these are harmless, but some may cause disease, and for the vast majority, we don't know which. Good afternoon, I'm Diego Letres. I'm a PhD candidate in the Sanchez Rivera lab at MIT, where my research focuses on understanding which and how genetic variants drive disease with a focus on cancer. Cancer is characterized by the abnormal growth and invasion of our cells, and it's driven by different genetic alterations. Understanding which variants drive disease matters because different cancers will respond to different treatments. And understanding which mutations drive cancer can lead to better targeted therapies, and targeting the right mutation can mean the difference between a therapy that works and one that doesn't. This is the goal of precision medicine. But this goal depends on understanding what each mutation does. Most genes encode proteins which fold into specialized 3-dimensional structures, and these structures determine their function. This framework is central to how we understand modern biology and how we interpret genetic variation. A genetic variant may change the protein sequence, change its structure, and disrupt its function. So a mutation in the wrong place can change the protein sequence, protein structure, and lead to dysfunction in a gene that carefully regulates cell growth, making it not able to do so or to actively drive cancer. However, a large fraction of the proteome does not actually fold into a stable 3-dimensional structure, and these are called disordered proteins. And for such proteins, the link between their sequence and their function remains poorly understood. So these regions are key to cell function, and they're frequently mutated in cancer, but we lack a framework for understanding what these mutations do, and this leaves a significant blind spot for cancer genetics. To address this, we turn to base editing, which is a precision gene editing tool that's based on a CRISPR-Cas system, where a guide RNA guides editing enzyme to a specific region in the genome, which allows us to model the mutations that we observe in human tumors. But the real benefit of this is scale. We can make libraries of thousands of these guide RNAs, and in a single experiment, we can model thousands of these mutations at a time and monitor their effects in human cells. Using this approach for disordered proteins, we've identified many different genetic mutations that significantly alter the growth and fitness of human cells. Each dot here represents a variant in a disordered region, and for most of these, they represent the first functional readout that actually links this specific mutation to a measurable effect. For specific genes, this is an example of cyclin D1. We can find where functional variants, where proteins harbor these functional variants. And this cyclin D1 is a regulator of the cell cycle. And we can see that these mutations cluster in a hotspot that is mutated in cancer. And this maps to a functional interaction that regulates these proteins levels in the cell. And disrupting this interaction promotes cell growth. We can find examples like this for many different proteins, highlighting that while some variants in structured regions can be interpreted through their changes in their structure, maybe some variants in disordered regions can be interpreted through the interactions that they perturb. But we really don't know all of these interactions, much less we know the structures of them. And while structure prediction has changed this for structured regions, they really fail to get a lot of information for disordered regions. However, a lot of these fold when they bind the right partner, and if we can find the right binding partner, we can model these structures and start to interpret what they do. So in collaboration with the Welcher Lab in Harvard, they've modeled over 1 million of these pairwise interactions, representing a large fraction of this interaction space. And integrating our functional data with their interaction maps across the proteome, we've identified many different interactions that may be perturbed by these genetic variants in regions and interactions that have not been yet structurally resolved. So taken together, putting these variants in a functional and instructional context may give us some insight into how they function in disease and hopefully what they mean for patients. Thank you. Thank you so much.
Good morning, everybody. And first of all, thank you so much to HEALS for this awesome opportunity to speak today. My name is Elijah Paivo, and I'm a 6th-year PhD candidate at the Institute for Data, Systems, and Society. Basically, from elementary school all the way up to starting my PhD, I was interested in robotics. I loved coming up with an idea, working through the engineering, and then finally seeing the robot move for the first time. But I began to wonder whether the engineering skills I had developed from robotic systems could be applied to societal systems to make an impact on a larger scale. That question brought me to IDSS, where I've been working on the US organ transplant system. Organ transplantation is a life-saving treatment for end-stage organ failure. Last year, in the US, 50,000 organ transplants were performed, and currently there's over 100,000 patients on the waiting list. When an organ is donated, it's actually treated like a national resource. That means that all of the compatible candidates across the country are ranked according to a policy called an organ allocation policy. And that uses factors like medical urgency, post-transplant outcomes, and things like transportation distance to decide the ranking. So they're all ranked. And these policies are established by an organization called the OPTN, with oversight from the federal government. And they have major implications for the national transplant system. They determine how many patients will die on the waiting list, how many years transplanted organs will continue to function, and even whether your chances of receiving a transplant might depend on how tall you are, what your zip code is, or what your blood type is. Okay, so these policies are made by committees of doctors and patients around the country. And when they make these policies, they start off by designing a few policy options and then evaluating them using computer simulation. Computer simulation predicts what the impacts of a proposed policy might be. The problem with this, it's one of the core, it's really the core piece of analytical tool for designing these policies, and yet the issue is that running one simulation can take nearly 7 hours. And to evaluate one policy, that requires 10 simulations. So this really bogs down the policymaking process and makes it very difficult for policymakers to really make progress. To receive a written report analyzing just a handful of policies can take 4 to 6 months. And this is a plot, and they just really analyze 10 policies at a time. So this really slow trickle information for these policies trying to make really complicated decisions makes it very hard for them to improve the system. Okay, so how do you make progress with this core analytical tool that takes so long? Well, this is where a little bit of MIT magic comes into play. Let's see. Yeah, okay. Well, so this is where a little bit of MIT magic comes into play, and my background in robotics. We were able to create a new algorithm that performs the same simulations, but much faster. Rather than taking 7 hours, it can now just take 15 seconds. So that allows us to do a lot of things. Let's see if this runs, if I hit play. First of all, instead of just simulating a handful of policies now, we're able to simulate tens of thousands. We can also use iterative optimization algorithms, what you're seeing here, to figure out what scores you should really be assigning candidates. So here, this was to figure out what scores we should assign to equalize access regardless of what your blood type is. And then finally, we developed, let's see if this works, we developed a interactive website that allows policymakers to design policies and assess trade-offs in real time, to try to replace those written reports that take so long. Okay, so we're really just at the early impact of this. I think there's much more to be done, but this is a plot that shows the annual number of patients removed from the lung transplant waiting list because they became too sick to receive a transplant or because they died. And in 2023, you can see that that number dropped by over 100 patients, or nearly 40%. That actually happens to be the year that the first policy that our tools helped design was implemented. Okay, so with that, none of this could be done without my amazing advisors and collaborators at UNOS, MIT, and the OPTN, and of course the support provided by MIT HEALS. If anyone's interested, I'll be at the poster session later today. Please feel free to find me and come talk. And thank you all very much.
Hello, everyone. I'm Casey Love. I'm HEALS Graduate Fellow in the Department of Biological Engineering, and I work in the lab of Professor Katie Galloway in the Department of Chemical Engineering. And in my work, I'm really interested in combining control systems and engineering principles with a mechanistic understanding of gene expression in order to both investigate and build molecular systems in mammalian cells. As a synthetic biologist, I look to program cells with new or enhanced functions by engineering them and rewiring them to sense internal and external cues, compute those diverse signals, and respond with complex cellular programs. To do this, we can deliver gene circuits, which are custom combinations of genes and regulatory elements that control the production and interaction of biomolecules that interface with the native pathways in the cell. And this allows us to leverage existing biology for new ends. These engineered cells have broad applications, both in basic research, such as designing circuits that can report on or perturb cell state, as well as increasingly in the clinic in the form of cell therapies or in vivo gene therapies. In this last example of a gene therapy, we envision delivering a replacement gene to correct for a mutation in a patient's genome, where the therapy would restore levels of a functional protein to treat disease. However, this simple approach has a number of challenges, including variation in gene dosage, which means that some cells don't express enough of the replacement protein, while others express at too high levels that can lead to toxicity. And indeed, when we deliver a simple unregulated gene to primary neurons, we see heterogeneous expression across the clusters of cells, with some cells having moderate levels and others having really high expression. So what we want instead is a gene circuit that can maintain consistent expression despite any variability in gene dosage. So with my colleagues here at MIT, I worked to build a circuit that does just this. On top of the therapeutic gene, we added regulatory elements that implement a motif known as an incoherent feed-forward loop using molecules called microRNAs. For more details on how this circuit actually works, come find me at the poster session later today. We combined this design with mathematical models and iterative testing to find one that worked well across diverse primary cell types, including the neurons you see here. And so in this image, you see clusters of cells having much more homogeneous expression without those high and potentially toxic levels. The next step for this circuit is to control expression of therapeutic payloads in relevant disease models to see if we can restore functionality. Gene therapy is just one example of a case where we need really precise expression in our engineered cells. I'm really excited to uncover the design principles that will allow us to build cells with these properties. And with the support of MIT HEALS and in my graduate work so far, I've used these insights to try to address several challenges, including dosage, which you heard about today. Ultimately, I believe that combining engineering with biology and bringing together expertise from both of these fields can help us program cells with much more precise and predictable expression that will enhance the impact of these technologies across the health and life sciences. So with that, I'd like to thank my advisor, Katie Galloway, my rest of my lab, the HEALS team, and my funding sources. Thank you.
Good morning, everyone. My name is Kunzan, and I'm a graduate student working at the Computational Biophotonics Lab at MIT RLE-ECS, advised by Professor Sixian Yu. And today, I'm very happy to share some of my recent works on label-free microscopy to study cell dynamics. As an engineering student, I was always amazed by how delicate a biological system is. All cells of ourselves, they need to take up oxygen and generate energy, but in very different and complicated ways that is part of our metabolism. So the metabolism is a set of chemical reactions that keep cells alive. It is converting nutrients into energies, building blocks, and signals that are controlling the cellular behaviors. So for example, cancer cells, they need to rewire their metabolism to grow faster, while immune cells, they need to change their metabolic states to combat with infection. So by studying the cell metabolism, we're really studying how cells make decisions, when to divide, migrate, or die. And fortunately, in this complex machinery, there are several cofactors that autofluoresce that can be probed with light, so that allows us to study them with microscopy. So for example, NADH and FAD, there are two metabolic cofactors, and this allows us to image them and understand the cells using these intrinsic readouts. So that's why we call it a label-free approach, because we don't need to label the cells, but instead we're using these endogenous contrasts. So my research goal is to develop and advance this label-free metabolic microscopy to study cells. And for example, this is a breast cancer microenvironment, and we can see the cancer cells. They're really proliferative, and they show high FAD signals. And we can see distinct colors of the cells because of their different metabolic states. And this metabolic microscopy can be even more comprehensive and powerful when integrated with structures. So we leverage the material properties of the tissue to visualize the extracellular matrices and also interfaces also in a label-free manner to provide context to the tissue environment. And so for example, now you can see fat cells, immune cells, and lipid vesicles. And a single modality only provides partial information of the cellular environment, and the integration of these multiple contrasts creates a colorful depiction that can reveal multicellular interactions. And this multiplexing resolves the complexity of a tissue. So for example, this is an intact mouse whisker pad tissue. This is unprocessed tissue, and was also previously in exhibition of Koch Institute Image Gallery. So with these different colors and also morphologies, we can identify different cell types, for example, stromal cells, muscle cells, and fat cells. And we can also see a complicated structure, a layer structure of this tissue, like basement membrane and epithelium. We can extract even more information of this multicellular interaction by leveraging the three-dimensional imaging capability of this technology. So for example, this is a precise characterization of a neuromuscular junction. So through the integration of these contrasts, this provides us opportunities to study these intricate dynamics inside the natural environment. And these contrasts are universal. That means that we can use these contrasts to study different organs and species with a fresh view, with rich structural and metabolic information. As an engineering student, my research goal is to further push forward the optical performance of the system in depth, speed, and resolution, and to use it to solve biological questions together with collaborations across the campus. And we aim to continue developing active tools that can combine optics, machine learning, and bioengineering, and to use them to identify, quantify, and manipulate cellular features that will ultimately enhance our understanding and strategies in diagnostic and therapeutics. And with that, I would like to thank my advisors and collaborators. Thank you.
Thank you to our fellows and graduate students for those inspiring, very inspiring presentations. our undergraduates are another essential part of what we're building here. My name is Katharina Ribbeck. I'm faculty in biological engineering, and I have the honor to introduce our undergraduate speaker today. Now, when undergraduates begin working in research, I often notice something. They ask questions that more experienced scientists sometimes stop asking. Not because experts aren't brilliant, they are, or can be, but somewhere along the way, as we become specialists, we learn to narrow the question. Undergraduates still ask, who is the patient we're trying to help? What is their experience of that disease exactly? And how might my discovery actually change their care? That instinct to keep the human in view is exactly what translation requires. And it is exactly what we want to protect and grow through the HEALS undergraduate research program. And over the past year, we have funded over 100 undergraduate researchers who are interested in translational science across 16 different departments, spanning life sciences, economy, social sciences. The interest in doing translational research was so strong that we, this fall, will be launching a formal clinical innovators track for undergraduates. And this program is the seed of something larger we're building at MIT, a formal pathway where MIT students move between laboratories, hospitals, and patients, not at the end of their training, but at the very beginning and throughout. So for today, we had hoped to have 2 undergraduate presenters, but one is currently in an exam, which is very much the reality of MIT life. You'll be able to meet him at the poster session, though, this evening, where a number of our undergraduate and graduate researchers will be sharing their work. We hope you'll stop by. Our other presenter is Janina Ojeduma. Janina, the stage is yours.
Hello. Good morning, everyone. My name is Janina Ojeduma. I'm a third-year undergrad studying bioengineering, and I'm a SuperUROP in the Engelward Lab, funded by HEALS. I'd like to present my research on genetically engineered cell lines for precision genotoxicity testing. Here's the clicker. To start, our cells face DNA damage from a variety of sources, anything from sunlight to inflammation in the body. My lab and my research has focused on toxic chemicals and their ability to cause DNA damage. In particular, a class of toxic chemicals called nitrosamines. These are found in environmental pollution and also can be contaminants in food and pharmaceuticals. Now, I'd like to take you all to Wilmington, Massachusetts. This community is about 30 miles north of here, and in this situation, one of these nitrosamines, NDMA, found its way into the water supply. This community then faced a spike in childhood cancer rates, like the case of Nicole Razzo, who was diagnosed with leukemia at just 3 years old. But Nicole is one of many people who were affected by nitrosamines in the environment, and NDMA is just one of the many chemicals that can have devastating effects. There are a couple of challenges to studying these nitrosamines in the laboratory. For one thing, they are metabolically changed once they enter the human body. Liver enzymes act upon them, and this does influence the toxicity response we observe in cells. So in our cell-based platform, we've chosen to use HepG2 liver cells supplemented with additional liver enzymes to ensure that we capture the full scope of metabolic activity and its influence on toxicity. Additionally, if we want to resolve damage on a biomolecular level, we want to identify the type of lesion, not just how much DNA damage is in the cell overall. To achieve this, we've leveraged DNA repair pathways, creating knockout cell lines that help us resolve the type of lesions we observe from toxicity via the repair trapping approach. So what actually happens when NDMA influences the cells is this lesion highlighted in red. While it may seem relatively small, it causes big damage in the long run, leading to mutations, cancer, breaks in the DNA. Cells possess specific pathways to repair this damage, and this is what we want to take advantage of. We know that cells that don't have these repair pathways are more sensitive to those specific types of damage, so we've created knockout cell lines that specifically show breaks in the DNA when this exact damage occurs in cells. In practice, what this means is we take human liver cells, treat them with chemicals, knowing that our enzyme supplementation also activates the responses that we see in the body, and then we electrophorese. This allows us to see whether the DNA is undamaged or whether it's damaged, causing the breaks in DNA that we can quantify using image processing. In my experimentation, I'll show you a bit of data soon. We can compare the normal cells in blue with the modified cells in red. And when treated with NDMA, we see that the red bars are significantly higher than the blue bars, displaying more DNA damage and confirming that NDMA does act via those small lesions I showed you earlier. For future directions, we hope to apply the principles of this assay to even more chemicals. Like I said, NDMA is just one of the many chemicals prevalent in our environment, in our food, present in many of the recalls affecting just ordinary patients taking heartburn medicine and pregnant women taking vitamins. We hope to examine this also through morphological changes using other microscopy approaches and expand to flame retardants, PFOAs, and other chemicals in our environment, as well as those that have not yet been brought to market so that we can understand their potential for toxicity before any person is exposed. I would like to acknowledge my various funding sources and give a huge thank you to the Engelward Lab, especially my PI, Professor Engelward, and my mentor, Dr. Vandana Singh. Thank you all.